Hello everyone, let's get started. Today we're going to be talking about the process of transcription. Let's take a minute first and review the central dogma. Remember that the central dogma is a series of 3 statements, and those statements tell us how the flow of information occurs in a living cell. They describe how the DNA molecule is used by the cell to create all of the traits that an organism has, both internal and external, physical and biochemical and behavioral, everything that makes an organism unique. The central dogma describes 2 processes, transcription and translation, and together those 2 processes make up what we refer to as gene expression. So, the central dogma tells us that DNA is transcribed to make RNA. RNA is translated to make protein, and it's protein that creates traits. Transcription is the first part of the dogma. It's a process where the cell is going to use the DNA molecule, that molecule that all living cells possess. It's going to use that DNA molecule to create another molecule called an RNA. The DNA molecule is considered the template, and the RNA is going to be produced from that template using an enzyme. And what the enzyme is going to do is pair up nucleotides to the DNA template in a complementary manner, as we've discussed, and create an RNA. Now remember from our discussion of the RNA molecule that RNA has a lot in common with DNA, but it has some important differences, including a different set of nucleotide bases. So instead of having A and T and G and C, which we find in DNA, RNA contains A and U and G and C. So there is no thymine in RNA. Uracil replaces it. So between the DNA, that's over here on the left, and the RNA over here on the right, we're going to see some different looking base pairing. Now, another thing to remember about transcription is this. All RNA is made from DNA through the process called transcription. We're going to be talking about messenger RNA today. We're going to be talking about the RNA that is made to create a protein. But there are other types of RNA, remember. There's rRNA and tRNA. There's even a type of RNA called small interfering RNA, and so on and so on. All of these types of RNA are created through transcription. That's why the central dogma doesn't say that DNA is used to make mRNA, even though it's the mRNA that will go on to be translated to make protein. Now, the specifics of the process of transcription are going to differ between prokaryotic cells, the bacteria and the archaea, and eukaryotic cells, just as we've discussed before. In bacteria, the process is just a little bit simpler than it is in eukaryotic organisms like humans. So what we'll do today is we'll use the bacterial cell as our model to talk through the process of transcription, and I will point out to you where the major differences are in eukaryotic cells. Now, when we discussed DNA replication, we said that a cell has to replicate its DNA every time it enters into the process of cell division. Transcription is different. Transcription does not involve the entire molecule of DNA being replicated or copied. Transcription involves a particular region of the DNA, a gene, being transcribed, being used to make an RNA. So, a cell is only going to perform the process of transcription when it needs a particular protein. And during the process of transcription, the DNA molecule is only going to unwind in the area that needs to be transcribed. With the DNA molecule separated, with the two strands separated, the template DNA becomes exposed. Remember, unlike DNA replication, during transcription, only one of the DNA strands is going to be used, and it's the one that has the gene on it. So, complementary RNA nucleotides or bases are going to be matched to the template DNA. The enzyme is going to create a phosphodiester bond to connect those individual RNA nucleotides together. And the enzyme that's doing this work is called RNA polymerase. RNA polymerase is an enzyme that needs an activated NTP. Remember, that stands for nucleoside triphosphate. It can't work with an ordinary nucleotide. It needs an activated nucleotide. And that activated molecule has two extra phosphate groups added onto it. This is the same activated version of the molecule that we saw when we discussed replication. Now this is a busy looking slide, but let's walk through it together. What they're showing us in this slide is an individual ribonucleotide being activated by having additional phosphate groups added onto it. So if you start on the left-hand side, what you're looking at here is just a generic ribonucleotide. You can see a sugar and a phosphate group and a base. Now, yes, technically this is a deoxyribonucleotide. I know that because there's no hydroxy group here, but we're going to pretend that it's a ribonucleotide. Now, with the energy from ATP, the cell is going to add two additional phosphate groups onto this nucleotide. Now it has become activated. Now it has the potential energy in it that the enzyme can use to create a phosphodiester bond. So now what they're going to show us is an existing strand. Again, we're going to pretend it's RNA. So there's two nucleotides already in this molecule, and the enzyme's going to add this one. Remember where it does that. It looks for the 3' hydroxy group. It looks for the hydroxy functional group that is jutting off of the 3' carbon in the sugar. Those two extra phosphate groups will be cleaved. They'll be cut off, and the energy that's released in that process will be used to build a bond, a phosphodiester bond between the incoming nucleotide right here and the existing strand. So we just built this. We just built this bond. Now this slide shows a different drawing of the same process. If you look up at the top on the left, you can see a double-stranded molecule of DNA in red, and this DNA has been opened up to produce what looks like a replication bubble, but it's not a replication bubble. This is an area of the DNA that is undergoing transcription. So we have a template strand on the bottom and a coding strand on the top. The template strand is the strand that's going to be used to make RNA. And they're showing us that RNA in yellow. Now remember, the RNA is going to be a complementary match to the DNA. And like all nucleic acid, like all DNA and RNA, those two strands, when they are bound together, are going to be anti-parallel to each other. So if the 3' end of the DNA is here and the 5' end is here, that means the RNA is going to be opposite. The 3' end of the RNA will be here and the 5' end will be here. Now if we take this little area right here and we blow it up, it looks like this. All right, so this is that yellow strand of RNA being built, and this is the DNA template. Remember, this is a part of the DNA that has a gene. There's a gene here. And remember what a gene is. It's a piece of DNA that has code to build a protein and regulatory code. So the RNA is going to be made in the area for the code that builds the protein, because the RNA is going to carry that code to the ribosome, and the ribosome is going to translate it. Now, they've drawn in all the details of the sugar phosphate backbone for us, and they've shown us where the bases are jutting off. So we have a C and a C and a T and an A and a G and another G. So these 3 nucleotides have already been added in, and here comes the next one. So the enzyme, the RNA polymerase enzyme, is going to match an A in the RNA with a T in the DNA. So here it comes in, it's been activated. It has 3 phosphate groups on it. And the enzyme is going to cleave the outer 2 phosphates off and use the energy to build a phosphodiester bond right here. And then it will bring in the next nucleotide, this one. It's going to match a G in the RNA with a C in the DNA. Sorry, it's actually this C. It's going to cleave off the outer 2 phosphates. It's going to use the energy from that broken bond to build a new bond, a phosphodiester bond, between the incoming nucleoside triphosphate and the existing strand. Now remember, we said an activated nucleotide in DNA is called a dNTP. That stands for deoxynucleoside triphosphate. When we're building RNA, it's just a nucleoside triphosphate, an NTP. If you compare the DNA polymerase enzyme that builds DNA and the RNA polymerase enzyme that builds RNA, they're actually quite similar. Both enzymes catalyze the formation of phosphodiester bonds between adjacent nucleotides. Both enzymes require an activated form of a nucleotide. Remember, we call those dNTPs when we're talking about DNA polymerase, and NTPs when we're talking about RNA polymerase. Both of these enzymes also can only build their nucleic acid in one direction. They can only build it in the 5' to 3' direction. In transcription, only one strand, though, of the double-stranded DNA is being used by the enzyme. And that's different from DNA replication. So, RNA polymerase is going to be making use of the template strand of DNA. The other strand is referred to as the coding strand. Now, the code for the protein, the information for how to build the protein, is found in the template strand. And that's why the messenger RNA will be made from that strand. The reason the other strand is called the coding strand is because once the mRNA is produced, the bases in it are going to match the bases in the coding strand, with the exception of the uracil-thymine exchange. Now think about what I mean. The messenger RNA is made from the template strand. So that means the bases in the messenger RNA are going to be complementary to the bases in the template strand of DNA. Well, the coding strand of DNA is also complementary to the template strand of DNA. So the coding strand and the mRNA are going to have the same sequence of bases, with the exception of the U and T swap. That's how the coding strand got its name.